Hello, grade 11 students. In this video, we're going to talk about the structure and the chemical composition of the DNA. The three questions that we're going to solve them during this lecture. First, what is DNA? Where it's located and in what forms? So first, what is the DNA? DNA, it's deoxyribonucleic acid. D for deoxynucleic acid, deoxyribonucleic acid, that contains the biological instructions that make each species a unique one. DNA is passed from adult organism to their offspring during the process of reproduction. So your DNA is a combination between your mother and your father's DNA. DNA is different from one person to another, so it's not 100% compatible. It is about 90 to 95% compatible in the case of identical twins. As we know, the cell is very small, and organisms, they have DNA molecules per cell. So each DNA molecule should be tightly packed. This packing is in the form of chromosome. So the chromosome is the package form of the DNA. The chromosomes, they are found inside the nucleus. So the nucleus contains the chromosomes that carry the DNA. So your DNA is composed of, it's containing the chromosomes. The chromosomes, they carry the DNA, and here they are found inside the nucleus, meaning that the DNA is located inside the nucleus and it's formed of chromosomes. So chromosomes, they carry the DNA. They are located inside the nucleus. So in what form? We will talk about this question. DNA is made of chemical building blocks. These are called the nucleotides. So the nucleotides, they are the building blocks. These building blocks, they are made of three different parts: the phosphate group, the sugar group, and one of four types of the nitrogen bases. To form a strand of DNA, the nucleotides, they linked in the chain with the phosphate and the sugar groups alternating. I'm talking about this. This is the nitrogenous bases. We have adenine, which is the A, thymine, which is the T, guanine, which is the G, and cytosine, which is the C. So A and T, they are complementary, and C and G, they are complementary. Here we have the sugar phosphate backbone, which is responsible for the packing. And here we have the DNA strand, which is a double helix form. So the DNA is a double helix form. So the four types of nitrogen bases, they are the nucleotides. They are adenine of symbol A, thymine T, guanine G, and cytosine C. The order or the sequence of these bases determines what biological constructions they are contained in the strand of DNA. For example, the sequence ATC GTT may code for the blue eyes, whereas the sequence ATC GCD. So here we have the difference at one of these nucleotides might instruct for the brown color. So each DNA sequence, as we know, each DNA sequence is formed of the instructions or contains the instructions to make a protein, is known as a gene. So each DNA sequence that contains instruction and code for a protein is called a gene. So scientists, they use the term double helix. So use the term double helix in order to describe the DNA's binding, two-stranded together. So the DNA occurs in the form of two strands, so it's double helix. These shapes, which look like a twisted ladder, it's like a ladder which is twisted, give the DNA the power to pass along the biological instructions with a great and very precise form. So this is the DNA form, and this is the double helix of the DNA. Here we have in the DNA, we have the hydrogen bond between the nitrogenous bases, phosphodiester bond, sugar phosphate back bond, and we have the 5 prime. In the second we have the 3 prime. So here we have C, cytosine, it's complementary with the guanine, A, adenine with thymine, guanine with cytosine, and thymine with adenine. So they are complementary, and we have the sugar phosphate backbone. Here are the chromosomes. These 2 chromosomes, they are homologous chromosomes. Homologous chromosomes. Here we have the centromere, which is the centromere. These are the 2 non-sister chromatids, where they are attached upon replication in the middle by the centromere. The 2 sister chromatids, here we have what's known by the P arm, short arm, and the Q arm, which is the long arm. And these together, they are called the sister chromatids after replication. Let's move to this application. Pneumococcus, a bacterium that causes pneumonia in mammals. Pneumonia is a disease that affects the lungs and exists as 2 types. Type S, which is a virulent strain surrounded by a capsule, and upon the injection of mice with this strain, pneumonia was observed and the death of the mice is followed. So S goes for the smooth. So I will repeat. Type S is a virulent strain surrounded by a capsule. Upon the injection of the mice with this strain, we inject the mice with this strain, which is the S strain, the mice die. And the death of the mice followed by after the injection of the S type. While type R is non-virulent strain that lacks a capsule. So R is for rough, while S is for smooth. So S causes the death. So in 1944, Avery and his team, they performed again these experiments, and the results are mentioned below in this graph, in this figure. So first, we have document A, 2 strains of bacteria, they have different feathers. So after injection of the S strain into the mouse, we have the death. While after the injection of the R strain, which is non-virulent, so the mice remain alive. So it stays alive. So meaning that it doesn't cause the death. So meaning that, first of all, the injection of the S, it causes the death. So type S is fatal, meaning that it causes the death. Whereas the type R, after its injection, doesn't cause the death. So it's not fatal. Okay? So this is the interpret for the first part. So after the injection of the type S strain of the bacterium pneumococcus into the mice, they die. What does this indicate? This indicates that the type S, the smooth type, is fatal. Whereas after the injection of the type R to the mouse remains alive. So this indicates that the type R is not fatal since it doesn't cause the death of the mouse. Whereas in document B, the experiment of Avery and his team, the bacterial transformation. So what did they do? After the injection of the DNA of the type S, so this is the DNA of type S, and the alive strain R, which is the non-fatal one. Okay, we inject the DNA of the S strain with the alive strain of R into the mouse. Okay, what happens? They die. So this means that the injection of DNA of the S with alive R strain, R plus some S derived from that transformation. So we have transformation from the R strain into the S, and this leads to the death. So whenever we have the DNA of the S and we have the RLA strain, alive RLA strain, they are injected together into the mouse. R strain, which is the rough, which is non-fatal, non-virulent, with the S derived from the transformation. So S from DNA from the S and the R strain, which is the non-virulent, it transformed into the S. This will lead to the death. So what can you explain this? This means that the DNA of the S, it transformed the R into the virulent S. So the DNA of the S changed the R strain into the S strain, which causes codes the death. Interpret this experiment. What can you conclude? So here we can conclude that the DNA is the carrier of genetic information and not the proteins. So DNA carries genetic information, since after the injection of the DNA of the S and the virulent R type, this will lead to the death, meaning that the R type is transformed into the virulent S type, S strain, and causes the death. So interpret, after the injection of the type S strain of bacterium Pneumococci into the mice die. What does this indicate? This indicates that the type S is fatal, while after the injection of the type R strain of bacterium Pneumococci into mice, the mice remain alive. So this indicates that the type R is not fatal, since it doesn't cause death. Document B, we inject the DNA of the type S strain and the alive strain R, rough strain. The mice die, even though the R is not fatal, it doesn't cause death, but we have the DNA of S. So what does this indicate? This indicates, and pay attention, the indication should be specific for the experiment. This indicates that the DNA of the S transformed the R into S. It changed from the R strain, which is non-virulent, to the virulent type, which is the S. Conclusion, we can conclude that DNA is the carrier of the genetic information, and only the DNA and not the protein. That's why the R strain transformed into the virulent S strain. And here we'll watch a small video talking about the structure of the DNA.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent the same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of four possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through two hydrogen bonds, and cytosine preferentially pairs with guanine through three hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single-ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms two repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.